I'm Peg McCarthy, for those who don't know me, and I'm the current chair of the Department of Pharmacology. And since Jay Magaziner couldn't be here today, who is the host of the seminar series, I was really honored that Edna asked me to read some remarks that she prepared, trying to encapsulate Edson in 2.5 pages, which is with a 16 font, which is impossible, but it's a lovely history of Edson. So Dr. Albuquerque received his MD degree from the Universidad Federal de Pernambuco and his PhD degree in physiology and pharmacology from the Escola Paulista de Medicina in São Paulo, Brazil. After earning his PhD degree, he received postdoctoral training at Tulane University and the University of Illinois under a Rockefeller Foundation Award, and subsequently at the University of Lund and the Karolinska Institute in Sweden. In 1968, Dr. Albuquerque was offered an honorary position as a Boswell Fellow at the University of Buffalo, New York. There he established his laboratory and was rapidly promoted to professor in 1972. So do the math. He went from a fellow to a professor in 4 years. And acting chair of pharmacology in 1973. So he went from professor to chair in 1 year. It was in 1974 that Dr. Albuquerque was recruited to the University of Maryland School of Medicine as professor and chair of the Department of Pharmacology. Under his leadership, the department gained international stature because of breakthrough findings of his own research program and those of faculty he recruited, many of whom are here today. The department also became known for the training it provided to postdoctoral fellows and to multiple generations of PhD and master's students through the graduate program in pharmacology that he established in 1974. In 2010, Dr. Albuquerque stepped down from the chairmanship in pharmacology and became the director of the Division of Translational Toxicology that he founded in the Department of Epidemiology and Public Health. Dr. Albuquerque's research centered around the cholinergic system and the peripheral and central nervous systems. He pioneered several electrophysiological approaches that enhanced the applicability of microelectrode techniques in terms of sensitivity and spatial and temporal resolution to the study of synaptic transmission. His contributions were noted for their creativity and conceptual developments. During his long and distinguished research career, Dr. Albuquerque published more than 400 peer-reviewed scientific articles with major contributions to the fields of both pharmacology and toxicology. In pharmacology, Dr. Albuquerque's pioneering research on neuronal nicotinic receptors laid the foundation for the discovery of the nicotinic allosteric potentiating ligands, of which the Alzheimer's drug galantamine is a prototype. In toxicology, his research has led to a better understanding of the long-term effects of organophosphorus insecticides and nerve agents and paved the way for the development of new antidotes. Dr. Albuquerque's scientific citizenship extended to numerous agencies and organizations. For many years, as chair of the Brain Disorders and Clinical Neuroscience Studies section and member of other study sections at the National Institutes of Health, and as members of committees in several agencies, including the Department of the Army, the Office of the Surgeon General, the National Research Council Board on Environmental Studies and Toxicology, and the Veterans Administration. Dr. Albuquerque dedicated time to the review of grants, research programs, and regulatory documents, guidances, and guidelines. Throughout his career, he also served the American Society for Pharmacology and Experimental Therapeutics, known as ASPET, organizing meetings and acting as field editor of the Journal of Pharmacology and Experimental Therapeutics. Dr. Albuquerque had a strong commitment to educate the next generation of pharmacologists, and his career was marked by his mentorship of hundreds of young investigators who have taken prominent positions in academic research, pharmaceutical industries, and governmental agencies in the US and abroad. He was a leader in international cooperation on the pharmacological sciences, particularly forming alliances with Brazil and European countries where he received much of his early training. The international training program in molecular pharmacology that he established between the University of Maryland School of Medicine and the Federal University of Rio de Janeiro in 1987 and directed until 2000 created a unique opportunity for graduate students from Brazil and the US to participate in exchange training at both universities. More than 100 American and Brazilian trainees, graduate students, and postdoctoral fellows have benefited from this program. As a result of his contributions to the fields of pharmacology and toxicology, Dr. Albuquerque was the recipient of numerous accolades over the course of his career. Those awards include the Order of the Grand Cross and the Rio Branco Award from Brazil, the Friedrich Merz Visiting Professorship from the Johann Wolfgang Goethe University in Germany, the Jacob Javits Neuroscience Research Award from the NIH, and the Otto Kreyer Award from the American Society for Pharmacology and Experimental Therapeutics, to name a few. Dr. Albuquerque was also a member of the Brazilian Academy of Sciences, the Latin American Academy of Sciences, and the Third World Academy of Sciences. And just to add on a personal note, I know many people in this room knew Edson very well. I had the pleasure of many stimulating interactions with Edson. You never walked away from a conversation with Edson and said, well, that was boring. He was always interesting. He was always fun. He was always curious. And it really has been my great honor to step into taking over the leadership of the Department of Pharmacology and the many wonderful faculty that he had recruited, and to build on his legacy to have great strengths in both neuro and oncopharmacology. So I'd like just to have a round of applause for Edson, who everyone misses to this day. Someone introducing Dr. Dingledine. Okay, all right, and now on to our speaker.
So thank you so much, Beck. Can everybody hear me? Well, okay. So thank you so much, Beck, for the introduction. It's always amazing to hear how much was done throughout the years, and to be a part of some of that history has been really an experience of a lifetime. I wish more of us had had the opportunity to interact with Edson as much as we all did. Some of us did. But it gives me great pleasure, and I'm absolutely honored. And so all of us who had worked with Edson and had known of his friendship with so many within and outside the university. So for everyone who is attending in Zoom, and there are many, and everyone who is in the room, I'd like to introduce to you Dr. Raymond Dingledine, who was a longtime friend, and he's professor in the Department of Pharmacology and Chemical Biology at Emory University School of Medicine, where he was former chair of the Department of Pharmacology and overlapped so much with Edson while Edson was the chair of department here at our university. So Dr. Dingledine received his bachelor's degree from Michigan State University in biochemistry in 1971, moved on to earn his PhD degree in pharmacology under Avery Goldstein at Stanford in 1975, got his postdoctoral training from Leslie Iverson and John Kelly at Cambridge between 75 and 77, and then under Per Anderson at Oslo between 1977 and 78. He then joined the Department of Pharmacology at the University of North Carolina in Chapel Hill in 1978 and rose to the rank of professor. And after spending a sabbatical year between 1990 and 91 in Steve Heinemann's lab at the Salk Institute, he then moved to Emory University in 1992 as chair of pharmacology. So there's something there about rising so quickly through the ranks. It's quite amazing. A job that he held then for 25 years. And for 11 years also served as active associate dean for research in the School of Medicine at Emory. He was editor of molecular pharmacology between 95 and 2000, again, that time when you both overlapped as Edson was in the board of JPAD. And he's currently on the scientific advisory board of Neurological, a clinical stage biotech startup that he co-founded. His career was profiled in Nature Medicine in 2002. And in 2010, he was elected to the US National Academy of Medicine. And in 2018, to the Norwegian Academy of Science and Letters. And then in 2019, he co-founded Paraffin and serves as chair of its boards of directors. Dr. Dingledine has co-authored more than 250 peer-reviewed papers and is co-inventor of at least 8 awarded patents. Since 1971, his research has been funded continuously by the NIH and other agencies. He was founding director of the Emory MSP graduate training program, and his trainees have gone on to leadership positions in academic, government, and for-profit organizations. So he's well known for his work on an NMDA receptor. So many of us have heard of his discoveries of the glycine site on the NMDA receptor and how glycine acts as a co-agonist of glutamate at those receptors. But more recently, motivated by the therapeutic opportunities that follow from discovering how the brain responds to injury, prolonged seizures, and gradual adjustments to aging, his research has focused mostly on the consequences of unabated neuroinflammation, particularly that mediated by COX induction and prostaglandin receptor activation. He and his colleagues have drawn on multiple disciplines to create a suite of novel, competitive, and allostatic prostaglandin receptor antagonists. They're potent, selective, brain permeant, and orally available. So these models of epilepsy, these antagonists are anti-inflammatory, and they provide a range of therapeutic benefits in models of epilepsy, head injury, arthritis, and endometriosis, to name a few. So his studies have demonstrated that EP2 mediate the prostaglandin-2 mediated inflammation is a profound driver of COX-2 related pathologies in multiple chronic disorders. It is with the great pleasure and great honor that I give the floor to Dr. Dingledine, who will be speaking to us this afternoon. And after this meeting, if you all join us for a short reception outside, you may have the opportunity to talk to him also in a one-to-one. So thank you so much, Ray. It's our pleasure.
Edna, thank you very much. I really feel, okay, thank you. I'm very honored to be here to talk on behalf of Edson. I wish he were here today. He was a remarkable person, as Edna mentioned. I've known him for many decades in a variety of situations and circumstances. And we had some good times together. The thing that I would, so I looked through Edson's CV and I picked out a few things that were particularly meaningful to me. These 4 papers are from different aspects of his career. Each of these very influential papers. You know, this one's still ongoing with Todd and the crew here at University of Maryland. This one's still ongoing, the galantamine. And as an antidote or a countermeasure to nerve agent exposure. That's the most recent situation in which Edson and I overlapped. We were both part of the Counteract National Consortium of Laboratories. He and his group developing galantamine. We developing anti-inflammatory compounds that I'll tell you about a little bit. But if you have a look, I mean, this shows his citation and publication history. The vertical lines are papers published per year. The solid blue line is number of citations. And this is how we would all hope that our careers would progress. Even as the number of publications was getting smaller, as he was focusing more on long-term issues rather than quick shot papers like we all do early in our career. But even so, the recognition of the value of these papers continued to rise. You know, as Peg mentioned, he's had a number of recognitions of his, for his activities. Of these, Otto Kreyer Award is, to me, a very, very special award within the US. As Peg mentioned, my PhD advisor, a guy named Avram Goldstein, Stanford, was a student of Otto Kreyer when he was a medical student at Harvard. And I heard many stories of Dr. Crayer and particularly his ethical approaches to life. That's a big component of anybody who gets the Otto Crayer Award, and Edson definitely deserves that. I looked through his students and was interested in how many of them I personally know over the years. Of course, these are his major research areas, and I am so happy and honored to be here to help remember this remarkable guy. Looking at his image right here brings it back. I mean, Edson was always a quite intense individual, as some of you might know, and did not suffer fools easily, but could carry out a conversation on multiple levels and a whole variety of topics. He was really a very special person. I would like to mention these two disclosures. I'm co-founder of two small pharma companies, both of which are in the access lane still. We haven't got a dime of revenue from either one of them yet, although there's a lot of personal debt involved in them. Neurop came about from my days studying NMDA receptors and AMPA and kainate receptor modulators. This is a clinical company now. We have our one internally developed compound go through phase 1 clinical trials. We're trying to raise money for a phase 2 trial in subarachnoid hemorrhage. Anybody wants to contribute, see me afterwards. I'll have a little table up here. And then a couple of years ago, co-founded Pyrophyn, which is a company developing novel anti-inflammatory drugs. Epilepsy and delirium are the status epilepticus are the initial indications. So, talking about neuroimmune pathways in the brain, these pathways exist and function under normal physiological circumstances. And they are, in my mind, they're hijacked during a neuroinflammatory situation. You've got blood-brain barrier that functions normally to prevent small molecules as well as large molecules from the blood and cytokines coming in from the blood. In a way, it protects the brain from this, but the blood-brain barrier can break down at the slightest provocation. A guy named Kevin Staley identified and described a peripheral inflammatory reflex that involves vagal nerve activation through nicotinic receptors controlling the macrophage differentiation and release of inflammatory cytokines. And that can go awry when the vagal nucleus is engaged. Of course, neurons can suppress microglial activation by CXCL3 or CX3CL1, I mean, activating CX3CR1 receptor on microglia, and that promotes their activation. We found, and others have also found, that activation of a receptor for prostaglandin E2 can increase the mossy fiber input to CA3. Here showing a normal EPSC and then or EPSP in this case, and then in the presence of 1 micromolar butaprost and a somewhat selective EP2 antagonist, you get a massive increase in that multi-fiber response. That's a presynaptic effect of EP2 receptors, presumably activated by prostaglandin E2 that is released in under normal physiological circumstances. So when we began this work, it was as an attempt to understand how the brain is converted from a normal brain to a brain with epilepsy. And what does this mean? Well, even stepping back a little bit, epilepsy, I like to think of it as a neurologic disorder characterized by a chronically reduced seizure threshold in 1 or more forebrain pathways. It's conventionally described as a situation that involves spontaneous seizures. To me, it's a bit of a tautology. It doesn't really give you any mechanistic insight or ability to go further. But so if I think about it as a way in a situation which seizure threshold is chronically reduced. And so if you buy that, like I do, identifying proteins that regulate the expression level or functional state of ion channels that control seizure threshold would give you an in or a therapeutic strategy to interrupt or attenuate epileptogenesis. And there are a number of processes that regulate seizure threshold. Synaptic plasticity, one of the big ones that has been well recognized. A number of compounds that can enter the brain when the blood-brain barrier is broken down can also promote, and neuroinflammation is a third mechanism. And so, or third mode, and our hypothesis that has driven work in my lab for the last 12 years or so is that an anti-inflammatory therapy could be disease modifying in rodent models of epilepsy, more recently delirium. And our targets have been cyclooxygenase 2 and CCR2, which is a cytokine receptor for CCL2. I'll describe that in a little while. The COX-2 began following a microarray consortium that I had organized, consisting of 11 institutions throughout the world, studying the 4 most commonly used models of epilepsy. Each model studied in 2 different labs, and we focused only on gene expression changes occurring in dentate granule neurons. And the idea was to come up with model-independent and lab-independent transcriptional programs that might be that are definitely involved or associated with and could be drivers of epileptogenesis. Out of that came cyclooxygenase 2. And if you think about, then I began thinking about, is there any information available about that would lead you to suspect cyclooxygenase pathways might be involved in the development of epilepsy? And I came across this Taiwan study. It's a post hoc study of 64,000 patients in their insurance database. And they divided the patients into 2 categories: those who did not have rheumatoid arthritis, those that did. Those that did have rheumatoid arthritis, the duration of treatment that they had been on NSAIDs. And what they did was they looked at the hazard ratio adjusted for all the normal things for developing epilepsy. And they found that this peripheral inflammatory disease, rheumatoid arthritis, was associated with a elevated risk of developing epilepsy, and that risk goes down the longer you're taking NSAIDs, which are cyclooxygenase inhibitors. Now, there's a lot of caveats to this study, but it does show that there is at least an association between a peripheral inflammatory disease and the appearance of epilepsy. And we've recently, unpublished studies have shown that rheumatoid arthritis models result in neuroinflammation in the brain. Secondly, here is an example of, let me see if I can, let me come back here. I'm going to move this down, try to move it around so you can see all the important things. Immunohistochemistry of cyclooxygenase 2 expression in mesial lobe that was resected from an epilepsy patient. This woman had had epilepsy for 39 years, finally had her temporal lobe resected, got better after that temporal lobe resection. Looking at her hippocampus, this is CA3 region, there it was loaded with neurons that are overexpressing cyclooxygenase 2. This also happens in rat models of prolonged seizures. And mouse models I'm not showing. But for these reasons, we were thinking of exploring COX-2 signaling in epilepsy. And I just highlight the risk factors, the reasons why we started down this pathway. So most of these risk factors for epilepsy are associated with an inflammatory response in the brain. And that's known to be mediated partly by cyclooxygenase 2, based on effects of selective COX-2 inhibitors. COX-2 is the inducible form of cyclooxygenase. There's a constitutively active form called COX-1. COX-2 goes up in selected principal forebrain neurons after seizures. And we had done a bunch of preliminary data that now we've gone so far beyond, we'll never publish this stuff, that also increased our interest in cyclooxygenase 2. So we have developed a number of tools to explore the COX-2 signaling pathways after seizures. Got 4 rodent models of status, 2 models of peripheral inflammation that involve no neurodegeneration. Got mice with conditional knockouts of COX-2 that are limited to those neurons in which COX-2 is induced following prolonged seizures. As I'll mention, we've developed selective inhibitors of key prostanoid receptor, EP2. We've got in vitro models of microglial activation, blood-brain barrier breakdown. And we've got conditional knockouts of EP2 in the myeloid cells, microglia and monocytes. Now, looking at these COX-2 pathways, COX-1 and COX-2, they're a very complicated pathway in the body and in the brain. You have a breakdown of phospholipids to arachidonic acid. That's converted by the cyclooxygenases to 5 prostanoids, including PGE2, and those activate 9 different prostanoid receptors. They're all G protein coupled receptors. COX-2 also is responsible for converting endocannabinoids to 8 different prostanoid-like molecules. Each of these has its own pharmacology. And so, the fact that we wanted to look downstream to get some selectivity. And we picked EP2 as a focus. And I'll talk about that a little bit later. So the first studies that we did, we knocked out COX-2 in the neurons that are, in which COX-2 is elevated following status epilepticus. And we found something initially that really intrigued us. One was that although there's no change in acute mortality, percent death during status, there's a great reduction in the delayed mortality that occurs within a week of status epilepticus. So knocking out this enzyme in a small neuron population in the brain reduces mortality, delayed mortality, following status. This was important clinically. There have been studies showing that in the 1 month mortality after status, after somebody's released from the hospital, 1 month mortality is somewhere between 24 and 39%. It's pretty big mortality after people have been moving around. We also showed that I won't give the details here, but we did a Barnes-Maze memory assay in these animals to understand, can they remember a hole in this Barnes-Maze table that they had learned where it was before status? Can they remember it 21 days later? And animals that have wild type animals and conditional knockout animals treated with saline had a perfectly good ability, in fact, a little bit improved ability to recognize that hole. But the animals that had received pilocarpine in this case and had status epilepticus had a deficit. They really could not remember where that hole was. On the other hand, the neural conditional knockouts behaved not differently than animals that had not seen status. So we, encouraged by that, we went to the cellular level and we first demonstrated that cyclooxygenase 2 is eliminated after in the CA3 region after status epilepticus. If you look very carefully, and you probably can't see it here, there are little tiny dots of COX-2 that we found are in microglia in the brain, but they're not knocked out by this neuroconditional knockout. This knockout was neuroprotective, both measured 4 days after status and 4 months after status. The number of Flora jade neurons per section was reduced over 10-fold in the neuron conditional knockout in the CA1 region of the hippocampus. 4 months later, doing Nissl stains, you can see, again, a reduction in the number of CA1 pyramidal cells. And the conditional knockout blunted that. Big, florid neuroinflammatory response to status epilepticus in wild type animals treated with pilocarpine is 4 days after status. Green is reactive astrocytes. Red is reactive microglia. And they are, when you set the imaging parameters and have it identical in between all of these conditions, you see that there's a big micro and astrogliotic response to status epilepticus. On the other hand, the conditional knockout, you get a much blunted response, and that is primarily limited to the cell body layer in this case, in the dentate gyrus. We looked at blood-brain barrier breakdown as assessed by albumin penetration into the into the cortex. And in wild type pilocarpine treated animals, you get a big albumin signal, which is greatly reduced in the conditional knockout animals. In fact, if you look overall, there's no induction. So we were interested then in the cyclooxygenase pathways that were responsible for the reduced mortality, reduced neurodegeneration. And we thought EP2. Why EP2? Well, COX-2 inhibitors, how many of you, probably some of you anyway, are familiar with the Vioxx history. This is a compound developed by Merck, a terrific anti-arthritis drug, really good, effective, caused heart attacks. Merck withdrew it. They withdrew another COX-2 inhibitor. There's now only one on the market, Celebrex, which has a black box cardiotox warning. And there will, in my mind, never be another COX-2 inhibitor introduced in the market. So we looked downstream, and the EP2 global knockout reduced inflammation and was beneficial in animal models of Alzheimer's and multiple sclerosis. And that suggested to us that maybe EP2 was a pro-inflammatory receptor. If you look at these, the COX-2 makes these 5 prostanoids. The cardioprotective effects are mediated by PGI2 and thromboxane. The gastroprotective effects are mediated mainly by EP1 activation. And we have found that EP2 activation mediates all or most of the inflammation that is caused by COX-2 elevation. Now, when we started, there were no EP2 antagonists available. So we thought, well, let's make our own and test them out in animal epilepsy models. And so at that time, I had co-founded a high throughput screening facility in our department. And we decided to make use of this. First, we created a cell line that expressed human EP2 receptors, and we created a live cell assay in a 1536 well plate. EP2 is a GS coupled GPCR, so when it's activated, it elevates cyclic AMP. We screened 262,000 compounds for their ability to inhibit PGE2-induced cyclic AMP accumulation. Out of those, we got 656 hits. They were scattered mostly throughout the plates. This is in our diversity library. We had a secondary assays for selectivity and false positive assays. 16 passed, which gives you the sense of how many false positives you get in a primary screen. Of those 16, we repeated the dose response for EP2. 13 were confirmed. These 13 were in 3 chemical classes. And we decided to explore this class initially. We explored the other 2 subsequently. And our first stroke of luck in this project was that we had EP2 antagonists that had potencies between 2 and 20 nanomolar in our diversity library. In the diversity library, we had compounds that were so potent as EP2 antagonists. And as part of this high throughput screening facility, I had been involved in 26 other projects that came into us nationally. Nobody had anything better than 1 micromolar. So we just lucked out. And so the problem with this molecule was that it didn't get into the brain and it had a very short plasma half-life. We did a little bit of medicinal chemistry, we meaning Thoda Ganesh, substituting a trifluoro for a methyl group right there. And that came up with a much improved molecule. This molecule was a competitive antagonist of EP2, as demonstrated by a Shield analysis. You know, here's a PG2 dose response curve, and then in the presence of increasing concentrations of the antagonist, you get a parallel shift to the right. Shield plot is linear with a slope of nearly 1. So the Shield KB is 18 nanomolar. It's a pretty, it's a nicely potent compound. We repeated this. We made cell lines for all the other prostanoid receptors and looked at the Shield KB to look at, initially it looked at selectivity. They were all this compound was quite selective for EP2 over everything but DP1, in which it was about 10-fold selective. Turns out the half-life is about 1.5 hours in mice and rats. Brain to plasma ratio about 1.6, 1.7. It's orally available in mice and rats. So it's a pretty good molecule, good enough so that we could do some in vivo animal work. Subsequently, Thoda and his group made, well, now I've got 450, now it's up to about 600 compounds, 600 analogs of the original, with the idea of getting, here's the 100-fold selectivity marker, and we were looking at EP2 Shield KB over the DP1 Shield KB. We've got compounds that are, differ by 1,000-fold in their EP2 potency. Now we have compounds that are way potent for selective for EP2. And our goal was to make compounds that are a little more aqueous soluble and more selective, which I'll talk a little bit about in a bit. So overall then, this compound, TG6-10-1, we felt had adequate selectivity, 10-fold over the nearest other prostanoid receptor, had decent brain PK, so we could use it for in vivo studies. And the first couple of questions we asked were whether blocking EP2 beginning after status epilepticus had been terminated, does that recapitulate the effects of ablating COX-2 in those forebrain neurons? We measured delayed mortality, weight loss, neurodegeneration, et cetera. Secondly, could any of the effects of EP2 block be due to reduced intensity of status epilepticus? If so, we could pack our bags and go home. And one thing I didn't mention was the cyclooxygenase 2 neuron conditional knockout. There was no diminution of the strength of status epilepticus as measured by EEG and behaviorally. Anyway, the answers to these two questions were yes, the effects of status are pretty much totally recapitulated by EP2 block and COX-2 neuron conditional knockout, and there's no effect on intensity of seizures. And what this means is that EP2 mediates most of the deleterious effects of neuronal induction of cyclooxygenase 2. I'm going to give you just a little, some of the experiments that showed this. And again, I'm going to move this thing around. Let me move it over here. Here's a study, days measuring mortality beginning the days after pilocarpine status. And here we gave the EP2 antagonist 3 times and then just let it go. And you can see the antagonist was protective against mortality. If you look at hippocampus and for inflammatory markers 4 days after status in the pilocarpine treated animals with post-treated with vehicle, you get a very intense astrogliosis in red, microgliosis in green this time, which is diminished by the antagonist. Its fluoro jade indication of neuronal injury is greatly reduced by the antagonist. Here's number of fluoro jade sections or cells per in the hilus per section. It's reduced by about half. Blood-brain barrier integrity, which is really diminished after the integrity itself is, demolished after status epilepticus, as indicated by albumin penetration into the cortex. Animals treated with the EP2 antagonist had very little, if any, albumin penetration into the cortex. So we wondered then, what was responsible for the neuroinflammation that we found? And I have to step back and say that by that time, we had done a number of studies with different status epilepticus models, different EP2 antagonists. And the one thing that was reliable in the way of neuroinflammation was an anti-microgliosis effect. Astrogliosis was diminished in some, but not all situations. So we focused our attention on the IBA1 positive myeloid cell population in the brain. And we meaning Nick Varvel, who was then in my lab, created a mouse that expressed GFP behind a microglial specific promoter. These mice had green microglia, and he crossed them with a mouse expressing red fluorescent protein behind the CCR2 promoter. And CCR2 is expressed by monocytes, but not microglia. CX3CR1 is expressed by microglia, but not monocytes. And what you see is in the saline-treated control brains, you get you can see the microglia. In pilocarpine-treated animals, you see swollen microglia as expected from microgliosis, but also now the invasion of monocytes from the blood. And in other studies I'm not showing, we identified CCL2 as a cytokine that is released from the brain, activates CCR2 on the monocytes, draws them into the brain. Now, CCL2 is expressed by microglia and by perivascular macrophages. It goes up very quickly after status, but the monocytes take a few days to come in. Don't really know why yet. But we asked, IBA1 will stain both the monocytes and microglia. And question we had is, which of these cell types is responding to EP2 inhibition? Nick Varvel had joined my lab. He's a neuroimmunologist, came from a German lab. And he showed that the CCR2 knockout mouse pretty much eliminates the infiltration or invasion of the brain by circulating monocytes after pilocarpine or after kainate. In this case, he looked at these monocytes that are marked by RFP in a heterozygous mouse, and you see a lot of monocytes in the CA3 region in both pilocarpine and kainate, but either in the global knockout of CCR2 or with a CCR2 antagonist, you get way diminished numbers of infiltrating monocytes. At the same time, he demonstrated that circulating monocytes themselves are much more pro-inflammatory, at least in terms of the cytokines that they express, than either resting microglia or activated microglia. He had pulled them out by flow cytometry from brains. You know, when Nick started these experiments, I thought, Nick, you're wasting your time. We've already shown in cultures that microglia respond to EP2. And it looks like these IBA1 cells are microglia. You're just wasting your time. But as it turns out, Nick was onto something. One way that this might come about is if you are preventing the release of monocytes from the bone marrow, either by a CCR2 antagonist or CCR2 knockout. And that's not the case. Lymphocytes, neutrophils, monocytes, they were all, not very different in the blood. After status epilepticus, either in saline treated animals or, as I'm showing here, animals after status epilepticus treated with vehicle or antagonist. Now, we found that by making blood smears and looking at the nucleated cells, the white blood cells, staining them for CD11B, which is a myeloid marker, and EP2, that we could find a proportion of myeloid cells also express EP2. And in fact, 24% of the CD11B positive cells were also EP2 positive, whereas 9% of the CD11B positive cells were EP2 negative. In other words, 77% of the myeloid cells express EP2. And following that, Nick did an experiment in which he asked, if you block EP2 receptors with our TG6 small molecule, what will that do to monocyte infiltration in the brain? And what he found was it greatly diminished monocyte infiltration. So what that says, let me come back to that in just a minute, but just hang that in your mind for a minute. Blocking EP2 systemically prevents monocyte infiltration in the brain. Nick found that monocyte infiltration after status is pathogenic, both by using the antagonist and, in this case, the CCR2 knockout. There's neuroprotection, blood-brain barrier the rigidity is maintained in the after pilocarpine by the knockout and also by the antagonist. The antagonist accelerated weight regain in these animals. Here I'm looking at, now I'm going back and forth, I know, but the main point is that both the CCR2 knockout and the CCR2 antagonist are neuroprotective and preserve the integrity of the blood-brain barrier. And so, contrary to my initial thought, and as so often has happened in my lab, I tell somebody, you know, it's don't waste your time, and they waste their time, it comes out good. So people are learning something from that and about how to deal with me in the lab. But at least part of the beneficial effects of the systemic EP2 antagonist or the knockout is very likely due to preventing brain infiltration by the monocytes. And I'm not going to, for time reason, I'm not going to talk about the myeloid cell EP2 knockout, but I just wanted to look at showing there are similar effects of the of COX-2 conditional knockout, systemic EP2 antagonist, myeloid conditional knockouts of EP2, and the CCR2 knockout or the antagonist across a wide variety of outcome metrics after status epilepticus. And this bottom one, spatial and working memory deficits after status, is, in my mind, really important. Cognitive comorbidities of people with epilepsy and after status epilepticus are major and greatly impact the quality of life. In fact, there was a survey done about 5 years ago now, people with epilepsy, and the question was, what is your biggest challenge? So number one was knowing when the next seizure is going to happen, timing of the next seizure. But number two were the cognitive fuzziness that they felt. They didn't feel that they were back to their normal, mental abilities. We have spent quite a bit of time looking at memory assays in models of status epilepticus. And here's an example of an experiment. This was done when Edson and I were both in the Counteract program. Here we are producing status, we meaning Shibo Rojas, producing status in rats, and then with disopropylfluorophosphate, then treating them with either vehicle or an EP2 antagonist, starting 2 hours after status, treating for 2 days. 2 to 3 months later, we carry out what's called a novel object recognition test, where we allow the animals to familiarize themselves with 2 identical objects. 2 hours later, we replace one of them with a novel object. And a normal rat or mouse will spend a lot more time with the novel object because partly because they're curious, but they also remember the familiar object. And we have a discrimination index. Here are how animals that don't undergo status or TG6 perform very well in this. They are easily able to discriminate the novel from the familiar object. Those that had status but vehicle were behaving at random performance. They had no ability to identify the novel object. And here the animals that had been given our EP2 antagonist for 2 days and then tested 2 to 3 months later were basically were able to identify the novel object. And if you think about it, there are 3 proposed causes of cognitive failure after status and also after Alzheimer's. One is neuronal death. You're just disrupting the circuits that are responsible for memory. Another is neuroinflammation. You are functionally changing those circuits. And then a third is aberrant neurogenesis, which has attracted a lot of attention lately, but in my mind has just not had a definitive test. Our work argues against neuroprotection as a primary driver of cognitive preservation by EP2 antagonists. And that is, we have three arguments for that. We have identified an EP2 antagonist, TG1177, that has a perfectly normal anti-inflammatory effect in the brain and eliminates this memory deficit, but has no effect on neurodegeneration following status. There are a couple of other models of peripheral inflammation that induce neuroinflammation with no neuronal injury. And they're also associated with a spatial working memory deficit that is totally restored by one of our EP2 antagonists. Finally, Tally Barham's group has developed rodent models of febrile seizures that have neuroinflammation, but no noticeable neuronal injury. And they also produce cognitive deficits. I'm only going to show one of these now. I've got slides for all of them, but the time is getting late. This compound TG1177 is, it improves delayed mortality after status epilepticus, like our earlier compound did. It is greatly reduces microgliosis throughout brain regions. I'm showing the amygdala. And in a Y-maze assay, the animals that, let me get rid of this again. Where am I going to put it? I'm going to put it right there. Animals that that do not undergo status epilepticus, minus or plus our antagonist, perform very well in this Y-maze assay of spontaneous alternation. Normally a mouse or a rat will, if he starts in arm A, he'll go to B, and then next he'll go to C, because he hasn't explored that most recently. And so you can quantify their behavior in these alternation assays. And animals that have had status epilepticus, but vehicle, are performing at background levels. They don't know where they've been. So that's considered a model of spatial working memory. Whereas animals that have been treated with our antagonist are behaving, performing just like normal. And if you look at fluorojade positive cells per section, the antagonist does not diminish those at all, even in a dose that's quite a bit higher than is anti-inflammatory. So I'm going to skip Twin Shan's data and just jump to a conclusion slide. And we have a model of what's going on here. And that model is that status induces COX-2 in a population of neurons in the brain. That releases PGE2, which activates EP2 on microglia, and that promotes the activation of microglia, which we have shown in microglial cell cultures. Also promotes release of CCL2, which, as I mentioned earlier, draws monocytes into the brain. And weeks to months later, these monocytes differentiate to activated macrophage-like, microglia-like macrophages. Our hypothesis is that the reason for cognitive failure is that these activated macrophages prune excitatory synapses excessively. We've got some evidence for that with Western blots of synaptophysin PSD95. They go down after status and are brought back by our antagonist. We're currently using an IMERIS program to quantify the density of PSD95 inside microglia in these brains. And so two different conclusions. One is that COX-2 related morbidity after status is largely driven by EP2 receptor activation. And brain infiltrating monocytes are highly inflammatory and are responsible for at least some of these morbidities. Right now, our work is driving toward asking, where are the relevant EP2 receptors? Are they mainly on monocytes or are they on activated microglia? You know, the temporal issue between very quick release of CCL2 and a delayed infiltration of the brain by monocytes. Where do the monocytes come from? They come from the marrow of the long bones or the skull. This is an interesting one to me. Does EP2 control the CCR2 pathways that are responsible for, the chemo-attractive function of CCR2 activation? And can we develop this compound up through IND and towards the first clinical test of a prevention trial for cognitive decline after status epilepticus? We're doing this now as part of a study that began in that counteract program that Edson and I worked on, and we're continuing. So I just want to mention, here are the folks in the lab right now. Here are alumni in our lab that had contributed to this since about 2010. Ashibo is an instructor, Nick is an assistant professor, Thoda is an associate professor, and these, we now share a lab. We have a really vibrant, it's a great laboratory. And, but it's definitely a team sport. Okay, thank you. So with that, I will stop. I'd be happy to continue this ad nauseum, but I assume that many of you are, have other things to do tonight. Tonight being Halloween. Anyway, thank you very much. And thank you for helping to remember this guy who's added so much to the field of pharmacology.
So I think we have a few minutes for some questions. If anyone has any questions. Tom?
So I have a couple of questions. First is, are there downsides to wanting the microglia activation?
Yeah, so that's a very interesting question. And what we have found is that if you start blocking EP2 receptors too early, there's no effect and a slight exacerbation of neurodegeneration and neuroinflammation. So like in many situations, at the beginning, inflammation probably plays a beneficial role. But then as it goes on and is not abated, it turns, turns bad. And so we delay blocking EP2 receptors for between 2 and 4 hours after the onset of status. The COX-2 is induced within 30 minutes. So you get a couple of hours of PGE2 production, presumably EP2 activation, and then we begin inhibiting. So that's in a way, that's going to be really beneficial for our intended indications. Following status epilepticus, these patients are, under the weather. They're being monitored intensively. They're being given all kinds of drugs. And then several hours later, they would be amenable to enter the trial. So you had another question.
That's fascinating. The other question is much broader. Obviously, there are many sort of information component, and I was curious whether you explore these same small molecules.
Well, yeah. Yeah, we have and are asking the questions whether EP2 antagonists are active in rat models of rheumatoid arthritis. Answer is yes. They reduce the arthritic pathology. Animals are up and around more. Of delirium, like post-operative delirium. And the answer again is yes. Post-operative delirium is something that is not acknowledged by many, particularly surgeons, but it happens, and our antagonists are beneficial in that regard. Others have used, and we're also studying a couple of models of Alzheimer's disease and showing that the antagonists diminish moderately, not as much as after status, but they diminish the inflammatory features that are coming up in these mouse models. Others have studied endometriosis with our molecules, and endometriosis is a highly inflammatory disease of the peritoneum. Peritoneal fluid is full of PGE2, and our antagonists, one antagonist that was tested reduces the size of endometriotic lesions and reduces endometriotic pain, and that's a big one. Hardly any pharma company wants to approach endometriosis. They just don't want to do it, but it's a problem of 10% of women of childbearing age worldwide. It's a big problem. There's a Japanese lab that have used EP2 antagonists in models of aortic aneurysms, and they find that the aneurysms are shrunk by EP2 antagonists. So there's as you might expect, COX-2 is a major inflammatory driver in a number of chronic inflammatory diseases. And EP2 apparently is part of many of these, not all. Thanks for that comment. There are other. Okay. All right.
I have a quick question. Is the EP2 the primary driver of opening the blood-brain barrier? Or is that, and is that independent of the stimulation of CCL2 production?
Don't know. I mean, that's what comes first. What we know now is that EP2 activation promotes blood-brain barrier leakage. We know that both from our in vitro models and from the effects of the antagonist and the myeloid conditional knockouts and the COX-2 neuroconditional knockouts of COX-2. We don't know.
Is the leakage big enough to allow the monocytes in, or is it extravasation occurring in addition?
I don't know that for sure, but the blood-brain barrier is not a single entity, and extravasation of albumin very likely depends on different pathways than cells infiltrating through the barriers. But so I don't know the answer to that. It's a really important question.
So trying to wrap my head around everything, but if I understand correctly, antagonize the CCR2 to prevent this infiltration. So is the receptor or microglia then not really a factor? It sounds like monocytes are really driving things. And you know, if you were able to give it to antagonize that 3-day window when cells have already infiltrated the brain, seeing the effect.
Very interesting question. Don't know. We're thinking that because of the summary that you just gave, that monocytes are responsible for at least part of the inflammation that is prevented by EP2 antagonists. And I have to say that quantitatively, the response to CCR2 antagonist or knockout is quantitatively as strong as the response to EP2 antagonist. So it may be that the primary target is monocytes. So that's one thing. Secondly, if you FACS sort monocytes have invaded and microglia from the brain, even activated microglia, you find that EP2 levels in monocytes are more than 10 times higher, RNA levels anyway, than they are in microglia. So monocytes have a strong ability to signal through EP2. And we're now purifying monocytes from blood and studying them acutely to understand their EP2 signaling pathways, primarily whether they use protein kinase A or EPAC pathway. The microglia primarily use an EPAC pathway to drive inflammation. But you know, these are good questions that will, you know, maybe we don't need a brain permeant EP2 antagonist, for example. All right, well, thank you again. Thank you very much. I'm saying hello to this guy.
Thank you so much for attending today. If you join us for a short reception outside, I just would like to present Dr. Dingledine with the plaque on behalf of the department and the School of Medicine. Thank you so much. We really appreciate it.
Wow. Thank you very, very much. I'll hold this up in front of this. Really, really something. I appreciate it.
Yes.